Hello, dear student. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, dear student, we are going to talk about today about the experimental proof of semi-conservative mode of replication. In previous class, we have already talked about the process of this replication, how DNA replication occurs, what are the enzymes used and how it takes place. That all we have discussed. Today, we are going to talk about the experimental proof that is given by a scientist for the semi-conservative mode of replication. So here, in this semi-conservative mode of replication, when replication occurs, one strand of DNA conserved as it is of a parent, where one strand gets synthesized new. So the DNA replication by a semi-conservative mechanism, and this was experimentally proved by Matthew Messelson and Franklin Starr. So it is also known as Messelson and Stahl experiment, and which is done by the use of E. coli. Okay. Now, when the experiment done in experiment, if we talk, what they did first. So he performed experiment by using heavy isotope, heavy nitrogen, that is in E. coli. So he performed experiment by using heavy nitrogen, that is N15 in E. coli. So this experiment performed by Messels and Stahl and by using a heavy nitrogen, that is N15. So first, what they did, if we talk, they grew E. coli in a medium E. coli in a medium containing NH4Cl where nitrogen is of N15 type, heavy isotope. So they grew E. coli in a medium that is containing NH4Cl as the only nitrogen source as the only nitrogen source for many generations. And N15 is the heavy isotope nitrogen. So N15 get incorporated into newly synthesized DNA. Into newly synthesized DNA as well as other nitrogen containing compound. So this way they grow into the N15 medium. So if we look over the experiment, what they did, in first they grown the particularly petri dish that having a NH4Cl and NO15 type, and they grow a bacteria. Now bacteria when grow and it replicates in its DNA, it has a nitrogen containing nitrogenous bases. So both the strand get labeled as N15. But when this bacteria is shifted to the medium for the next generation, which containing NH4Cl and that is of N14 type, then this both strand get this, you can see red color, they are N15, they are separated, and this red color is going to be a what, a N15 type, and this blue color is going to be N14 side. So it is a N14 N15 hybrid. Same hybrid is obtained in the another strand because these two strands get separated. This is N15 and this is also a N15. But new blue strand get is synthesized, that is a N14. So it is a N15 N14 hybrid are obtained. Then again for the next generation also, if we go on growing them in the NH4Cl containing medium where nitrogen is not a heavy, it is a light isotope, and when we grow a bacteria, then in a N14 medium, this then these two strands of this hybrid get separated, and this become a N14, and new strands will be synthesized, that is of N14 type. And here the particular red color is N15, and newly synthesized strand will be of N14. Here also in this also, if these two strands get separated, one become N15 and another become N14. So newly synthesized strand going to be N14 and N14. So such a way here this are the N15 N14 N15 N14. So these are the hybrid, and this is the N14 N14. So this heavy DNA molecule, which is the heavy isotope, could be distinguished from the normal DNA by a centrifugation. Normal DNA have a N14 isotope. So it is separated by a centrifugation. And here by a cesium chloride density gradient centrifugation. Which centrifugation is here? Here. So it is centrifugation by cesium chloride density gradient centrifugation. And in cesium chloride density gradient centrifugation, based on gravitational force, the particular DNA is going to be settled down. So the dense solution of this cesium chloride, which is present in this test tube or a tubes, okay, on centrifugation forms a density gradient bands of a solution of a lower density at the top, where that increases gradually towards the bottom with the highest thickness. Means the dense material will be settled down at the base, and the particularly low density medium material will be settled down on the upper side. So then they transferred the cell into a medium with the NH14, which we have seen, okay, and the cesium chloride density gradient has been measured. So here you can see N15 is a heavy isotope, so N15 band will be observed at the bottom side. Whereas N15 and N14 is a hybrid, so its band is somewhat on the upper side compared to the N15 and 15 because they have some low density. And when N15 and 14 isotope these are in the last generation, then they are at the same site in the previous one, but N14 and 14 isotope are at the upper position. Okay, like this, by the separation of density also, we can conclude that the particularly DNA is here replicated in a semi-conservative manner. So here they grew E. coli in a medium containing NH4Cl first, that is N15 is a heavy isotope, as the only nitrogen source for many generation. Then the result was that N15 was incorporated into the newly synthesized DNA as well as the other nitrogenous containing compound. This heavy DNA molecule could be distinguished from the normal DNA by a centrifugation in a cesium chloride density gradient, just we have discussed. N15 is not a radioactive isotope, be remember. It can be separated from the N15 based on the densities only because it is a dense isotope. Now then they transferred the cell into the medium with the normal NH4Cl. They took samples at various definite time interval as the cells multiplied, and they extracted the DNA that remained as a double-stranded helices. The various samples were separated independently on a cesium chloride density gradient to measure the densities of the DNA. And thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, That is after 20 minutes. One next generation will be obtained in a 20 minutes in the E. coli, because this E. coli divided in 20 minutes had a hybrid or intermediate density. So DNA extracted from the culture after another generation, that is after 40 minutes, that is second generation, was composed of a equal amount of hybrid DNA and light DNA. So by this, it was concluded that the parent strand is getting conserved and the one strand get newly synthesized. So increase in the amount of light DNA and decrease in the amount of DNA can be possible due to the semi-conservative mode of replication. So in prokaryote, the semi-conservative model is proved by the Messelson and Stahl by using a E. coli. But Taylor et al. They have proved semi-conservative mode of replication semi-conservative mode of replication in eukaryotes by using tritiated thymidine, that is H3 thymidine in roots of Vicia faba or faba beans. So it is by a Taylor et al. And Taylor et al. Proved by in eukaryote by using the roots of Vicia faba by using this tritiated thymidine in the eukaryote. Ken was one of the another scientists semi-conservative who proved semi-conservative mode of replication in E. coli by using tritiated thymidine, that is S3 TDR in autoradiography experiment. And he proposed theta model of replication in circular DNA. In circular DNA, they proved this theta model of replication. Okay, so that is about the experimental model which proved a semi-conservative mode of replication. Okay, so dear students, thank you for this.